In this video, we'll cover physical maps. When you think about genome maps, this is probably the map type that you're thinking about. Furthermore, as a professional, this is probably the map type that you'll interact with most frequently, because the backbone or base layer of a physical map is a nucleotide sequence. Unlike cytogenetic maps, whose base layer is the physical structure you observe under a microscope, or in the case of genetic maps, which we'll cover next, are based on recombination. We strongly associate genomes with A's, T's, C's, G's, and even U's. And a physical map shows the exact arrangement of the nucleotides that compose a genome, because a physical map is, after all, a nucleotide sequence. Compared to the other map types, cytogenetic maps and genetic maps, physical maps provide the most detail and have the greatest resolution. By resolution, I mean the ability to distinguish two things next to another. Let's say I have a physical map and a gene sequence, which is also a physical map, albeit a tiny one. I can compare the gene's sequence to the physical map to determine exactly where the gene occurs. But notice that a physical map provides nucleotide level resolution. I can determine the borders of a gene are defined by this G and this T, but the flanking nucleotides C and A are not part of the gene. Or to think about it another way, the gene is here, and these are sites where the intergenic spaces stop and start. This is what I mean by the resolution of a physical map. A physical map provides single nucleotide resolution, or the ability to associate a particular nucleotide with a specific feature or function. Cytogenetic maps don't provide this level of resolution. We can see a white band in a chromosome in a classical cytogenetic map and assume that there are probably expressed genes in that region. However, that white band might correspond to about 100,000 base pairs of DNA, which in no way provides nucleotide level resolution. Even when we step up to modern cytogenetic maps, we don't get single nucleotide resolution. We can generate a fluorescent probe that will detect a particular gene, but the lit up area might correspond to 10,000 base pairs due to the compaction of the DNA and resolution of the microscope. But the gene is really only 121 base pairs. The point is that physical maps of the three map types provide the greatest resolution, single nucleotide resolution, because physical maps are nucleotide sequences. In and of themselves, a genome's nucleotide sequence is pretty much meaningless. Imagine I give you a file containing the human genome physical map, all 3 billion plus base pairs. What would you do with that? Well, you'd probably go cross-eyed after gazing at sequences for a few hours, but beyond that, all you'd probably be able to say is that it looks like a random string of A's, T's, C's, and G's, and every once in a while, you'll come across a sequence that seems a little different. That's not particularly useful or informative. So when we talk about the power and utility of physical maps, we're not talking about the nucleotide sequence per se. Instead, we are referencing the annotations on a physical map. You can annotate a physical map with anything, literally anything, that directly or indirectly has something to do with an organism's genome. And it's these annotations that give physical maps such fantastic detail and power. We can annotate physical maps with all of the genomic features we've discussed, from genes to intergenic spaces to origins or replications, transposable elements to tandem repeats, including telomeres, centromeres, PAR sequences, and microsatellites, because all of these things are ultimately nucleotide sequences that occur at specific loci within a genome. But there are other types of annotations that you'll find in physical maps, ranging from the big, broad, and ill-defined to the super specific. Let's start with the big, broad, and ill-defined. You don't have to know the exact nucleotide sequence of something to annotate it on a physical map. For example, you can annotate bands from a classical cytogenetic map onto a physical map. Recall that we can measure chromosome length in micrometers. Well, that also means that we can measure band length and the distance between bands. There are conversion factors that allow us to approximate the number of base pairs in 1 micrometer of condensed DNA. So we can look at this black band and say that it's roughly corresponding to nucleotides 100,000 through 300,000. Because this band represents about 200,000 base pairs of highly condensed heterochromatin, and it's not on the ends, nor is it a waist-like constriction, then it's possible this area contains transposable elements. So we could go to our physical map, highlight region 100,000 through 300,000, and add an annotation, saying this area likely contains elements associated with ultra-compacted heterochromatin, like transposable elements. This information could be useful for someone interested in transposable elements, like me, and lets them know to check out this area. Now, this annotation doesn't provide single nucleotide resolution, because in reality, the black band could correspond to nucleotides 87,654 through 328,123. However, this is still a type of annotation that can be put on a physical map, even though it corresponds to larger regions with ill-defined borders. Furthermore, this is a putative or hypothetical annotation. There is no biochemical or genetic data establishing that transposable elements are in that region. However, the area may contain transposable elements based off what we know about ultra-compacted heterochromatin. The point is, we can annotate a physical map with putative or hypothetical assignments even if they have ill-defined borders. Now let's talk about super detailed annotations. Let's say you're a biochemist and you're all about understanding the properties and functions of protein X. Through many experiments over many years, you've figured out that these amino acids are involved in targeting the protein to a particular region in a cell. In contrast, these amino acids are involved in substrate binding. These amino acids are involved in catalyzing the reaction. But these amino acids are involved in binding to another protein. All of these amino acids are encoded in the nucleotide sequence of a gene. So you can go to a physical map and find the nucleotides that correspond to these groups of amino acids and annotate them, such that these nucleotides are annotated as a localization signal. These nucleotides are annotated as being involved in a substrate binding, whereas these nucleotides are involved in catalysis, and these nucleotides are involved in forming a protein complex. In this case, the annotation in a physical map isn't merely the nucleotide sequence of a gene, but instead, this gene is annotated with features that break it down into different functional groups. So physical map annotations can be crazy specific and ascribe functions and properties to particular nucleotide sequences within a single genomic feature, whether it's a gene, origin of replication, transposable element, or a tandem repeat. So as you can see, anything and everything that is directly or indirectly related to an organism's genome can be annotated on a physical map. This is why physical maps have become the foundation for many biological databases. We use physical maps as scaffolds to aggregate and organize everything we know about genomes and their features. It doesn't matter if the data came from biochemistry, cell biology, molecular biology, genetics, evolutionary biology, ecology, or an organismology field. Any information, regardless of where it comes from, if it some way relates to a genome, can and is annotated on a physical map. Let's look at one of these organismal databases that are based on a physical map. This is EcoSic, a physical map database for E. coli. These databases normally have some kind of genome browser that lets you look at the physical map and its annotation at different scales. So here's the genome level view. In this view, you don't see the nucleotide sequence that's the map's backbone. Instead, you see a bunch of annotations represented by different symbols. At this scale, this type of representation is common. The nucleotide sequence is not shown, but often the nucleotide coordinates are. If you zoom in on a position in the map, you restrict your view to smaller and smaller regions. And as the magnification increases, so does the resolution. In this view, you can see the nucleotide coordinates, and the line represents the nucleotide sequence. You can see the annotations more clearly and see annotations that weren't visible at the genome view. In EcoSiC, genes get colored symbols like purple, orange, yellow, brown, etc. Since genes are annotated on one of the two strands in a double-stranded genome, and at this level the genome is represented by a single line, it's the direction of the arrowheads that indicate each gene's strand annotation. By convention, arrowheads facing right mean the genes are annotated on the top or forward strand. Arrowheads facing left mean the genes are annotated on the bottom or reverse strand. Eventually, you can zoom into the nucleotide sequence level. You don't see as much of the genome, but the detail is phenomenal. You can see exactly where the genes start and stop with single nucleotide resolution. And at this level, the nucleotide sequences of both strands are shown. Every database is different in terms of its organization and how annotations are symbolically represented. However, since a physical map is a nucleotide sequence of one or two strands, physical maps adhere to standard annotation conventions. In a double-stranded nucleic acid, genes are annotated on one of the two strands, and replication origins, transposable elements, telomeres, centromeres, PAR sequences, and microsatellites are annotated on both strands. Typically, the annotations are hyperlinks that will take you to pages that tell you about that locus, its function, if known, and the scientific literature that relates to that specific locus. As you can see, physical maps are the base layers of databases that allow us to aggregate and organize everything we know about genomes and the functions and properties of genome features. And that's pretty cool. But the awesomeness of physical maps doesn't end there. Physical maps have given rise to a field called bioinformatics. Bioinformatics combines biology, computer science, information engineering, mathematics, and statistics to deal with the acquisition, storage, analysis, dissemination, and interpretation of biological data. Bioinformatics is a hot field and is directly and indirectly responsible for many of the most significant biological discoveries in the last 30 years. One of the real powers of bioinformatics is using computers to search for and find information. I'm sure you're familiar with a Google search, or even the find function in Microsoft Word or Adobe Acrobat. You enter a word or a phrase, and then these search and find algorithms go through a database or file and tell you anywhere that word or phrase appears. These are pretty powerful and useful functions because they tell you where to find the information you are most interested in. But words and phrases are just strings of characters, just like nucleotide sequences are strings of characters. So, like you can search the internet for Amazon, you can search a physical map for the sequence TGAC. These bioinformatics search algorithms will tell you exactly where that sequence occurs in the genome, and if it appears multiple times, the distribution of those sequences across the genome and how many times it appears. But like other search algorithms, bioinformatics search algorithms will allow mismatches and wildcards. So, you can set your mismatch preferences, and instead of just returning all instances of TGAC, the search function will tell you where AGAC, CGAC, and GGAC occurs. Now, let's take a minute and think about the power and utility of being able to query physical maps for particular nucleotide sequences or closely related nucleotide sequences. For a gene to be useful, it has to be expressed. On their own, genes aren't functional molecules. They don't do anything. A gene is nothing more than the information specifying how to make particular proteins or non-coding RNAs. Like we'll talk about in a few weeks, expression of a gene requires proteins and non-coding RNAs to bind to the locus, unwind the DNA, and convert the information encoded by the gene into an RNA. But these gene expression factors need signals that tell them where genes are in a genome and where to start and stop expression. These signals are nucleotide sequences. Now let's say you figure out the sequence motif that is recognized and bound by gene expression factors to start expression. You can then take that sequence and use a search algorithm to tell you where else in the genome that sequence or similar sequences occur. Why would this be useful? Well, now you've just generated a hit list of loci that could encode genes. This data in and of itself isn't particularly strong. It doesn't mean that every single locus in the hit list encodes a gene, but it gives you a place to start to try and identify novel genes in the genomic nucleotide haystack. The point is, once we learn the function of a sequence, we can search a genome for other instances of that sequence or similar sequences, and thus point us towards particular regions that could have specific functions. So we can search for sequences within a genome, but you can also search across genomes, and this is where things start to get really exciting. When we look at nature, we see all these different organisms that have very different morphologies. People don't look like trees, or birds, or salamanders, or fish, or mushrooms, or amoebas, or bacteria, or viruses. You may think that these extreme morphological differences must be due to drastic differences in their genome's nucleotide sequence. Well, that's not true. There are indeed differences in the nucleotide sequences in these genomes. However, there are also many nucleotide similarities. These similarities exist because all life is evolutionarily related. When organisms reproduce, they transmit their genomes to the next generation. Over time, genomes can change, but they also retain many of the features of their ancestors, which is why we find differences and a whole bunch of sequence similarities between genomes. For example, humans and the nematode C. elegans look different. There are nucleotide sequence differences between humans and C. elegans genome. However, about 80% of the loci associated with human genetic disorders have a direct parallel in the C. elegans genome. We share these nucleotide sequences because we are evolutionarily related. We call sequences that share identity or similarity due to the duplication events or shared ancestry as homologous sequences. Oftentimes, homologous sequences have the same or similar function, despite their occurrence in different parts of a genome or in different organisms. Because of homologous sequences, we can take the information that we know about the sequences of one organism's genome and apply it to other organisms' genome. This field is called comparative genomics, and it is very, very, very powerful. For example, we can take the nucleotide sequences for a gene expression factor and search for that sequence in other genomes. In this case, we're not trying to find possible genes in the organism that we're working with. Instead, we're trying to find genes in organisms that we haven't worked with. Through comparative genomics, we can take everything that we know about the structure and function of nucleotide sequences in one organism to mine and ascribe function to other organisms' genomes. You may have heard or read stories about how researchers found a gene in the human genome that does blank. Sometimes that information comes from hardcore biochemical or genetic analysis of that gene and its products. However, many times, the functional assignment doesn't come from investigations of the human gene or product. Instead, we figured out something in mice, found the homologous sequence in humans, and then ascribed the mouse gene function to the human gene function. And more often than not, this translation or transference of information turns out to be appropriate and valid because the sequences are homologous and the functional properties of the loci are retained. This takes us to the power and utility of model organisms like E. coli, a bacterium, Saccharomyces cerevisiae, a yeast, C. elegans, a nematode, Arabidopsis thaliana, a plant, Drosophila malangaster, a fruit fly, and Musc musculans, a mouse. These are the organisms that we've done the most intensive and extensive biochemical, cellular, molecular, and genetic investigations on. We take what we learn about their genome structure and function to annotate and ascribe function to the genomes of organisms that we haven't or can't study in great detail. And all of this is possible because of homologous sequences and comparative genomics. So one of the great strengths of physical maps is the types of comparative studies that they facilitate. We can annotate a physical map with almost anything and everything that is directly or indirectly related to genomes. We can then use these sequences and annotations to mine other physical maps and ascribe functions or possible functions to homologous sequences. In addition to taking a particular nucleotide sequence and searching for its presence in other genomes, we can also use comparative genomics to do something called de novo discovery. In de novo discovery, we do the reverse. We compare the nucleotide sequences of different organisms to identify nucleotide sequences that may have biological significance. If we compare 3 nucleotide sequences and see that a lot of it is different, but in all 3 genomes we find the exact same or very similar sequence, then that can mean that the sequence has some particular and important function. Through the process of mutation, genome sequences change over time. However, nucleotide sequences that control some biological function, like genes, tend to mutate at a much slower rate, and sometimes don't mutate at all. It's not that mutation in these sequences isn't possible. Instead, changes to those sequences are often not tolerated and don't persist within a population. So, seeing the same sequence or a very similar sequence in different organisms, especially distant related organisms, can indicate biological significance. It's through de novo discovery that we found new classes of genes, RNAs, and proteins, especially non-coding RNAs. Before we move to our next topic, I just want to unpack the ideas of de novo discovery a bit further. Seeing short sequence identities or similarities is common. We can find the sequence ATCG numerous times in a genome and in different genomes. And statistically, we would expect this. In DNA, there are 4 possible nucleotides, A, T, C, and G, meaning that at any one position, there can be one of 4 possible nucleotides. To figure out the number of times you would expect to see a particular sequence due to random chance, all you have to do is take the number 4 and raise it to the length of the sequence you are interested in. If we're interested in ATCG, a sequence that's 4 nucleotides long, then we would expect to find this sequence by chance once every 4 raised to the 4th times, or once in every 256 nucleotides. But if we are interested in a sequence that's 6 nucleotides long, then it would occur by random chance 1 in every 4 to the 6th nucleotides, or once in every 4,096 nucleotides. But genomes are way bigger than this. So once you get up to the 20 to 30 nucleotide length mark, then you start to get statistical significance, meaning that the chance you would find an identical sequence that is 20 to 30 nucleotides at random in your average genome gets pretty slim. This is why de novo discovery searches normally have a lower limit. We don't get excited about 4 nucleotide matches, but 50, 100, 1,000 nucleotides? Now you may be onto something. In the rest of this video, I will tell you about how we build physical maps. We can build a physical map for any nucleic acid that is part of any genome. We can generate physical maps for viruses, bacteria, archaea, and eukaryotes, including protists, fungi, plants, and animals. We can generate maps for nuclear genomes, mitochondrial genomes, chloroplast genomes. Furthermore, we can generate physical maps for both RNA and DNA, regardless of its strandedness or form, as well as every kind of double-stranded DNA that we've discussed, from chromosomes to mini-chromosomes to chromids and plasmids. To generate a physical map, you start with a sample of the organism's genome. To do that, you have to extract the genetic material from a pool of viruses or cells. There are many different extraction methods that tend to be specific for the organism, meaning that the extraction method for a plant is different from an animal, bacteria, or a virus. Once you have purified samples of the genome, the next step is to sequence it. We can sequence any genome regardless of its type or strandedness. The ability to sequence nucleic acids came around in the early 1960s, and the first physical map was the nucleotide sequence of a transfer RNA, or a tRNA. Yes, that sequence, all 75 nucleotides, is a physical map, because a physical map is just a sequence, regardless of size. It wasn't until 1995, however, that we had the first physical map of a genome, in this case, a bacterium. The following year, the first physical map of a eukaryote nuclear genome was published. Since then, the number of partial or complete physical maps of genomes has exploded. This is mainly due to advances in DNA and RNA sequencing technologies and supercomputers that can handle, process, and analyze massive data sets. There are several different nucleic acid sequencing technologies out there. We're not going into the specifics on any one method because the general trend is that one technique will be the gold standard for a while. Then a new chemistry and/or technology will come around and it will become the new gold standard until the next thing comes along. But the general theme in sequencing advances is cheaper, faster, longer, and better sequence reads. By sequence reads, I mean sequence fragments. Sequence reads are getting longer and longer. As of now, the longest sequence reads are at about 100,000 nucleotides in length. However, most people are using technologies that generate sequence reads that are a few hundred to thousands of nucleotides long. Now think about that for a second. The nucleic acids that make up genomes vary widely in size, but some of the larger nucleic acids, that being chromosomes, can be hundreds of millions of base pairs in length. And in the case of a segmented genome containing multiple chromosomes, the entire genome can be hundreds of billions of base pairs. Maybe you're beginning to see the issue. We can't generate in a single read the nucleotide sequence of most genomes. Instead, we sequence the genome as a series of relatively short fragments. Fragments that then have to be reconstructed into the full-length sequence of a nucleic acid. Reconstruction involves a method called tiling. Tiling requires fragments to have overlapping sequence at both ends. Let me show you how tiling works. Here we have a series of sequence fragments. We randomly pick one as a starting point or our reference. Then we look into our pool of fragments and find those fragments that overlap the two ends of our reference fragment. Through tiling, we take a reference fragment and build the sequence out from both ends by taking advantage of the overlap between fragments. Let's build a bit more. So at this point, we have a tiling array of fragments. I want to emphasize that we are not looking at double-stranded sequences. Instead, we are looking at a series of fragments that correspond to one strand. If we're dealing with a double-stranded genome, then the other strand would have a sequence that is complementary to this one. Once you've organized all your fragments, you can then write out the sequence of that locus, which in this case is this. So, as you can see, tiling requires that fragments have overlapping sequence. The overlap is critical. Without overlap, you would have no idea how to organize the fragments. However, with overlap, you can arrange the fragments appropriately. Tiling works well when you're dealing with unique sequences. However, tiling doesn't work for many tandem repeats, especially when the tandem repeat's overall length is greater than the fragment length. So you can understand what I'm talking about, let me show you an example. Here we have a series of sequence fragments corresponding to a tandem repeat locus. How would you build the tiling array? Is the real nucleotide sequence this, or is it this, or is it this? The answer is you don't know. You know there is a tandem repeat with this sequence motif, but you have no idea how long it really is. Is the tandem repeat 700 base pairs long, or is it 100,000 base pairs long? You don't know. Now there are ways you can figure this out. Still, the point is that accuracy of tandem repeat loci in a physical map tends to be wildly inaccurate because we can't necessarily determine the overall length of the tandem repeat. This means that regions in a physical map that correspond to telomeres, centromeres, PAR sequences, and even microsatellites can be very inaccurate. The sequence motif is there, but the overall length of the tandem repeat is often wrong. Here you could use the information from a modern cytogenetic map to improve these regions' accuracy. But cytogenetic maps don't provide the single nucleotide resolution that are the hallmark of physical maps. So these areas tend to be the genetic black holes in our knowledge about genomes. Another thing that I would like to point out is that physical maps are rarely full-length sequences of chromosomes, mini-chromosomes, chromids, or plasmids. Generally, the smaller the nucleic acid, the better the overall coverage. But a physical map is often fragmented when we get to large nucleic acids like chromosomes. In these cases, there are nucleotide sequences for some regions, but not others. Sometimes this is due to difficulties associated with sequencing certain areas that are oftentimes associated with highly structured areas of nucleic acids, like heterochromatin. But sometimes these biases are a reflection of decisions regarding cost, time, and goals. Oftentimes, researchers are more interested in possible gene spaces than areas that contain transposable elements, centromeres, telomeres, or whatnot. So the fragmented physical maps tend to be skewed towards gene spaces. The final point of our conversation about physical map construction is that the actual nucleotide sequence in the map generally comes from one individual or a population of clonal individuals. For example, the first draft of the Human Genome Map was Craig Ventnor's DNA. If you go into that database and see a T at position 103,456 in chromosome 1, that doesn't necessarily mean that you have a T at position 103,456. Chances are you probably have a different nucleotide at that position. The point is that the nucleotide sequence in a physical map is a reference sequence from one individual or a group of clonal individuals. It's not a composite sequence of a species genome. Though the total number of nucleic acids that appear in the physical map are a species composite. However, remember that we can annotate a physical map with anything related to a genome, including differences that we observe among individuals. So in our example, we can annotate position 103,456 in chromosome 1 with all of the different nucleotides we see in the human population at that position. So physical maps are also incredibly useful when it comes to aggregating and organizing the differences that we see between different individuals within a species. So that concludes our conversation on physical maps. You can generate physical maps from every lineage and every type of nucleic acid, regardless of its type, strandedness, or form. A physical map is a nucleotide sequence regardless of its size. We can annotate a physical map with anything and everything that is directly or indirectly related to an organism's genome. We can annotate loci as genes, origins of replication, transposable elements, and tandem repeats, including telomeres, centromeres, PAR sequences, and microsatellites. These annotations will tell us, with single nucleotide resolution in some cases, exactly where these features occur within a genome, their frequency and distribution across a genome. But we can also annotate loci with well-defined or ill-defined boundaries, with hypothetical or putative assignments. Furthermore, we can annotate a particular feature with its components. For example, we can annotate a transposable element with the location of the terminal inverted repeats, transposase genes, and spacers. Hence, of the three map types, physical maps provide the most detail and best resolution. Physical maps act as a platform for us to aggregate and organize all the information we have for a particular organism's genome. Using bioinformatic approaches, we can take what we know about the structure and function of a locus to identify where identical or similar sequences occur within a genome or different genomes. This allows us to transfer or extend our knowledge from one organism to another. Physical maps are generated by tiling sequence fragments. This approach works well for unique sequences, but doesn't work well for tandem repeats, especially determining tandem repeat length. Also, not all physical maps are full-length sequences of an organism's genome. Sometimes physical maps are nucleotide sequences of some, but not all regions of the genome. Despite these caveats, physical maps are the most utilitarian of all genome maps. They provide the most detail, have the best resolution, and can be annotated with anything related to a genome. Therefore, when you interact with a genome map as part of your career, chances are it'll be a physical map.